So I'm going to spend a few minutes telling you about how preclinical neuroscience in particular can inform us about our understanding of the neural bases of addiction. But I should introduce myself. My name is Tony Phillips. I'm the scientific director of the CIHR Institute that deals with neuroscience, mental health, and addiction. So what we're talking about today is really our bread and butter. But I'm also, you may know that all scientific directors also have to have a lab. So I'm delighted to be also a professor in the Faculty of Medicine in the Department of Psychiatry, where we do have an active lab studying this topic and others. Let's just get sure I got this right. So a few important assumptions that we really need to put on the table here. And these words haven't been said yet, and I think they really do need, we need to be reminded of them because they set the stage for what we're trying to understand. And of course that is the nature of addiction. And if we're going to try to model this on a biological basis, we need to be developing models that reflect the fact that addiction is very often reflecting compulsive thoughts and actions that are directed towards procuring and then self-administering drugs of a choice. Even when people go to great lengths to try to undergo detoxification and to rid themselves of these habits, sadly, it doesn't take very much to reinstate the behavior, so the strong tendency to reinstate drug-seeking behavior is a cardinal feature leading to the statement that addiction is really a chronic relapsing disorder. The addictive behavior can be induced by just the merest re-exposure to a drug, as often happens with nicotine addiction. Even stimuli, which we call in psychology conditioned stimuli that are paired with the action of the drug reward, if you get exposed to that, maybe it's the setting in which you took a drug, or maybe it's a reminder by way of an ad, that can be sufficient. And the word stress has been creeping into our discussions today, and so stressors can quite often be a predisposing factor to relapse. The last point is delving into the biological bases, which is what I want to get to, and I'm going to show you some evidence that suggests that perhaps relapse is mediated by an interaction of specific neurochemicals. The neurochemical dopamine and the neurotransmitter glutamate may come together and modify synapses, and that might form the basis for why addiction is persistent. The other point to emphasize is that now I think the incontrovertible evidence that all drugs of abuse, there may be one or two exceptions in some of the antidepressant drugs that don't act on the dopamine and glutamate systems, but every other drug that you can think of that people choose to abuse has a direct effect on brain neurochemistry. And what we now believe is that these chemicals, for reasons that we don't really understand, have the capacity to hijack the basic systems in the brain that underlie our capacity to be motivated, to have goal-directed behavior, and to experience pleasure. So the natural motivation and reward processes that have allowed us to adapt to a very complex environment provide a point of privileged access to many of these drugs. And not only that, and this third point I think is really a fundamentally important point to emphasize, the repeated exposure to these drugs of abuse has long-lasting modifications on brain structure and function. Art just introduced us to the idea that exposure to these drugs could be changing the way in which our genome is read. I'm going to give you some evidence that, not quite as a molecular basis, but the synapses in our brain are modified in a semi-permanent manner by repeated exposure to drugs. And therefore that the environmental stimuli and the direct pharmacological actions of drugs can be combined through learned associations, and that this may be an important determinant of addiction. So here's a slide from NIDA. This is the class of the high school students' art. So we're showing here just a very, very basic description of a synapse, which, of course, as you all know, is the site of the release of specific neurotransmitters that then act on specified receptor proteins and thereby generate new action potentials, electrical activity in the receptive neuron, and that's how information is conveyed in the brain. We know a lot now about the mechanisms that underlie modification of synapses, and I'm going to show you some evidence that repeated exposure to drugs like amphetamine and morphine and other drugs modifies the synapse. And we know how to interrogate this now with great fidelity. And so some very, very early work that my colleague Chris Fibberger and I did so long ago it's embarrassing. We employed a model. The other point to emphasize when we talk about preclinical work is that an animal, be it a rat, a mouse, a primate, every animal tested to date, even goldfish, will self-administer many of the compounds that humans choose to self-administer when given an opportunity to press a bar, which then activates an infusion pump and delivers the chemical directly into the vascular system. And this experiment really just began the field to start to think about the fact that catecholamine neurotransmitters were very important in this process, whether you were applying a drug directly to the body or stimulating the brain directly with electrical stimulation. Dopamine seems to be a critical neurotransmitter. We also did other experiments early on with animals, again showing that if you place an animal in a particular environment, and it can be randomly selected, it could just as easily have been that environment, and all the animal does is receive passively an infusion of morphine in this case, but it could have been cocaine, it could have been amphetamine, it could have been alcohol, it could have been nicotine, and as few as 4 pairings of the effect of the drug with a unique environment causes something called conditioned place preference. And what happens there is that following these brief exposures to the drug, you then put the animal in a choice point and let it indicate to you where it would like to be in this environment. And inevitably, the animals choose to go to the environment in which they received the drug. So this is a simple way of screening drugs to see whether or not they may have abuse potential. And this, what we did in this experiment, actually, this was done again in the dawn of time, shortly after we discovered, or we, but other people discovered that the brain had its endogenous opioid systems. And so there's something called metencephalin. And when we place metencephalin directly into the cell bodies of dopamine neurons, we could, in this paradigm, the animal would then choose to go to the environment in which it had received activation of the enkephalin system. So I'm just showing you these to convince you that we can use animals to tell us something about disorders as complex as addiction. In another series of experiments, we employed something called brain microdialysis that allows us to monitor changes in the brain dopamine system over long periods of time. The animals in this experiment were given access to amphetamine for 48 hours. And the animals avidly, this is the baseline lever pressing rate. Once that drug is available, the animal starts to administer the drug, and it continues to administer the drug for 18, 20, 24 hours. But it gets to a point where it chooses, and these are the lever presses here, it reaches a point where it abstains from any further drug self-administration. And we're monitoring dopamine in the brain throughout this entire 48-hour cycle. Through the period of active drug self-administration, the dopamine system is turned on immediately, maintained on throughout that active period. When the animal chooses not to administer the drug, it's had enough, immediately the dopamine system returns down to a basal level, and it's throughout this period, even we can give passive infusions of drug that will not reactivate the dopamine system, nor does it reactivate behavior. But after a few hours, we can give a passive infusion, or the animal can go over and press the bar itself. And when the dopamine system comes back online, the behavior comes back online. So these sorts of experiments have shown, and we can show that these things occur in specific regions of the brain. These experiments actually reinduced drug-seeking behavior by stimulating the glutamate pathway from the hippocampus to the dopamine pathways, thereby linking glutamate and dopamine together in this process. So I'm just giving you this sample of experiments from our own lab that are repeated thousands of times in many, many other labs throughout the world, telling us that dopamine and glutamate are critically important in determining addiction in animal models of behavior. So the take-home slide that we see in every textbook now points to the fact that in this lower red color here, there are dopamine neurons in the brain, actually relatively few. There's only around 5,000 or so in each side of the brain in the human, and yet they innervate vast areas of the striatum and also particular areas of the cortex and subcortex. And we know for a variety of experiments that that's the pathway that all drugs of abuse seem to activate, either by directly activating a dopamine neuron or disinhibiting it through an action on GABA neurons, or perhaps indirectly activating it through activation of glutamate. These pathways, it doesn't matter whether it's dopamine, opiate, nicotine, alcohol, this is the pathway of the brain that is activated by these drugs. And going back to an earlier slide, this is the pathway in the brain that determines motivation and reward-seeking behavior. Marco is going to walk us through some fascinating work with humans that show that the relevance of these generalizations from animals are very, very germane to the human condition. I won't preempt his talk, but by using brain imaging techniques such as positron emission tomography and fMRI and other techniques, you can show that these drugs of abuse are having a direct effect on the brain. And here's a classic study, one of the very first studies to look at this relationship between cocaine, the dopamine system, and even chocolate. This is the natural reinforcer turning on the dopamine cell bodies in the ventral tegmental area. And then here's work from Nora Volkoff and colleagues showing that you get similar activation with drugs of abuse. But Marco will go into this in much more detail. The final model of animal behavior that I want to emphasize in the remaining time I have is a phenomenon that applies equally to humans and animals, and it's called behavioral sensitization. And I said earlier that hallmarks of addiction are the craving, the thinking about, and the compulsive thoughts and actions, and also the fact that it's a chronically relapsing disorder. And we've been looking for neural circuits that might be involved in this craving behavior. And the field settled on a model called behavioral sensitization, which is manifested again in many different mammalian species. Most of the work is done on rodents. And what happens here is, as you'll see in the next slide, repeated exposure to a drug, rather than causing tolerance to the effect of that drug, actually leads to heightened reactions to the pharmacological effect of the drug. And in common with the development and maintenance of craving behavior, the effects of sensitization increase in magnitude with repeated drug exposure. And then even when the animal is given a drug holiday for weeks, if not months, these effects persist once they have been established, and you can re-trigger the behavioral correlate in rats of drug-seeking behavior with a single exposure to the drug. And some colleagues have suggested that what is maybe happening in addiction is that the dopamine system that we referred to a moment ago actually gets sensitized by the repeated exposure to these various classes of drugs, and that forms the basis of craving and possibly sets the stage for chronic relapsing disorder. And so this behavioral sensitization is shown in this cartoon. It's a 4-day period of time. This animal is receiving a single injection of cocaine, which is a stimulant drug, and it becomes a little more active. It also likes it, so it's a little more accepting of the injection on day 2, becomes more active on that day. Third injection on day 3, it's even more active. This is always the same dose of drug. And you give the 4th injection of the drug, the animal goes wild. And so somehow or other, 4 injections of this drug have modified the neural circuitry to the point that you get this very, very exaggerated response to the drug. And we call this behavioral sensitization. If we then imposed a timeout period of several weeks, and we come along on day, say, 30, and give another injection, we get this level of behavior. So once it's established, it's maintained. So what's the neural basis of this, and can it give us any insights into the neurobiologic basis of addiction? So we've been studying this for some time now, and one of the first things we did was to show that not only is the behavior sensitized by repeated exposure, but when we can do this microdialysis experiment where we can unobtrusively sample the brain and see whether dopamine is changing, what we get in the sensitized animals is a much stronger neurochemical reaction to a single dose of d-amphetamine than in animals that had saline previously. And this was another experiment that showed that animals that had been sensitized to repeated exposure, passive exposure to amphetamine, when we gave them an opportunity to self-administer the drug themselves, this is a trick where the animal has to keep pressing on a lever more and more times to get a single injection of drug, up to the point where they have to spend, say, 100 or more bar presses to get a single injection, as opposed to just a few initially. And this is called a progressive ratio schedule. They eventually reach a point where the work they have to do to get the reward the work is excessive relative to the reward, so they stop. And that's called the breaking point. And these are animals that weren't sensitized, so they're able to work, say, 25 times for each injection. Animals that were sensitized before giving access to amphetamine will work 5 times more. This is a log scale, so they're working about 125 times. We next moved on, so now we're trying to understand, we've established through our work and many, many, many other labs, the fact that the brain dopamine system is important. This is a motivational system, but we also recognize that environmental factors associated with drug abuse can be important determinants of the behavior. So another line of attack in our work at UBC has been to study the possible involvement of synaptic plasticity as a determinant of drug-induced behaviors. And so I'll introduce very briefly in a moment a concept called long-term depression of synaptic activity. And we have now, I think, some very, very strong evidence to suggest that it's this form of synaptic plasticity that may set the stage for the occurrence of behavioral sensitization, and this in turn is determined by the context in which the learning takes place. So what do we do? Sorry, where am I? This is now where I have to beg your indulgence. I do have another, the phenomenon of synaptic plasticity, which involves changes in either increasing the strength of glutamate synapses or decreasing them in the case of long-term depression, that we have the tools now to manipulate this process. Apologies for those slides missing. And so we know that the depression of this synaptic activity involves the removal of certain glutamate receptors called AMPA receptors. And my colleague Yu-Tian Wang at UBC has developed a technique whereby we can selectively block the removal of those receptors and thereby eliminate the occurrence of long-term depression. And so this has now given us a tool, a pharmacological tool, to affect these changes. And so what we've done is use this tool, and what we've been able to show is that if we give this, and we call this peptide, it's a glutamate AMPA receptor blocking peptide. This is one of the AMPA receptors. There's a sequence on the tail of AMPA receptors that need to be phosphorylated in order for this removal from the surface of the synapse to occur, and this blocks that. And so what we do in this experiment is either give saline plus that particular peptide or amphetamine in the peptide day 1 to day 5. the presence of that peptide doesn't block the initial change in locomotor activity. So these histograms are locomotor activity. This is the first time the animal got the drug. This is the fifth time. That is sensitization. The group that got this peptide that interferes with this AMPA receptor endocytosis still shows some initial sensitization, but the real acid test is what happens when you put them into a drug-free state for 2 weeks and come back and challenge with another dose of amphetamine. And so the peptide that we've developed is given coincidentally with the drug, as I said a moment ago, for 5 days. In the control group, when we bring them back in on day 24, last injection of amphetamine on day 14, they show a robust sensitization. They're showing much more locomotion. If we give a scrambled version of our interference peptide, same amino acids but in a different order, no effect whatsoever on the expression of sensitization. But if we come ahead and challenge again with amphetamine in the group that we'd interfered with AMPA receptor endocytosis, there's absolutely no behavioral sensitization. So by blocking this process of long-term depression, we think that we have a clue as to how we might be able to modify synaptic plasticity in a way that creates a pharmacological immunity, I guess, to subsequent effects of drugs. So needless to say, we're working very, very hard on this approach, and we think it holds great promise. So I hope I've been able to convince you that we have a variety of different animal models that allow us to interrogate the molecular bases of drugs' actions within the brain, and in particular those actions that have relevance to the study of addiction, that perhaps what drugs are doing is they're actually creating their own long-term memories in the brain, which are the factors that really regulate the habit associated with drug addiction. Or if Mary Olmsted was here, she would be emphasizing the impulsivity associated. This could be written into the synapses of the brain. So drugs of abuse can change brain structure by modifying synaptic plasticity, and we believe that one of the approaches to a pharmacological intervention that may be effective in treating addiction is to understand those mechanisms of synaptic plasticity and to modify them pharmacologically, and we have tools now that allow us to do that.